He's a very good colleague of mine. And he is also he's also a professor at New York Stem Cell Foundation, Robertson Investigator. And he earned his PhD from Imperial College London and carried the postdoctoral training at Harvard University. And he pioneered the first models of ALS using personalized stem cell-based approaches. His laboratory was established in 2015 at the Les Turner ALS Center here at Northwestern. And he uses the power of pluripotent stem cells to understand the biology and the pathology of motor neurons. And today he will be talking about ALS FTD and his recent research on ALS using the novel techniques and technologies that he has developed. Thank you so much.
Okay, good morning. Can everybody hear me? Can you hear me at the back? Yes, okay, great. So it's wonderful to be here again. I'd like to take the opportunity to thank Hyundai and Bob for the opportunity to present at this meeting and give you an update on some of the work that we do in my lab. And I'd also like to extend a thank you to the guest speakers, the people who have traveled to be with us today. And it's been nice to see, wonderful to see this conference grow over the years. So again, I'm delighted and grateful for the opportunity to be here and talk to you about the work that we're doing. So let's move this. So as I mentioned, what I'd like to do over the next 25 minutes or so is tell you a little bit about some of the work that we're doing in the lab. Those are my disclosures. So this is an ALS meeting, and I will not introduce the disease other than say that it is caused by the growing dysfunction and eventual degeneration of a very particular neuronal subtype called the motor neuron. Spinal motor neurons found in the brain, in the spinal cord, and the brainstem, and motor neurons found in the motor cortex. And we in the lab have been utilizing induced pluripotent stem cell technologies to try and get access to these patient-specific neurons to study disease processes. Now, as I'm sure most of you are aware, ALS is a highly heterogeneous disease in terms of its genetics. As I'm showing here, about 90% of ALS is of a sporadic nature, and about 10% of the disease runs in families. There's been tremendous breakthroughs in ALS genetics over the last 20, 25 years or so, and we now know of Mendelian variants in at least 30 genes that can cause ALS when mutated. And the fascinating thing about these discoveries is that these genes encode proteins that seemingly do a diverse array of cellular processes. And what I've done on this slide is I've simply categorized the genes based on the function of the proteins that they're associated with. And broadly speaking, there are three large classes of proteins. One's involved in RNA metabolism and translation, one's involved in proteostasis, and one's involved in cytoskeletal homeostasis and trafficking. And there's the odd genes like SOD1 and CHCHD10, which don't really fit any one of these patterns based at least on what the proteins, the encoded proteins, do. And one of the central question I've been trying to address in my lab in the 8 years that I've been here is whether there is mechanistic convergence between this distinct genetic subtypes of ALS. Is it one disease, or is it under the same umbrella, under the same clinical presentation, or is it different subtypes of disease? That's the question we're trying to address. And whether anything that we find out about all these rare genetic subtypes, does it apply, is it relevant for what happens in sporadic disease, which is, as I said, the majority of patients. And the way we address this question is we generate patient-specific iPSC models for each one of these genetic subtypes. And then we take a deep dive into the motor neurons that are derived from this iPSC model to try and understand how mutations in particular genes impact the function of the neurons. And in just a couple of minutes, I want to summarize a lot of work that we've done over the last few years to illustrate the point that whenever we study a specific genetic subtype, we always find, to this point, a connection, an interaction with another genetic subtype of ALS. So Andrew Fleming is a graduate student in the lab who's working on an exceptionally rare form of ALS, an ALS that's caused by mutations in a chaperone protein called DNAJCC7. And when Andrew tried to ask, what does DNAJCC7 do in human motor neurons, he found that it directly interacts with two other ALS causal genes, two proteins called matrine3 and FUS that are categorized under the RNA metabolism group. So Andrew is in the process of trying to understand the functional implication of this direct interaction. Another example that we recently published was work from a former postdoctoral fellow in the lab, Kostas Choras, who showed that VCP, which is a gene that when mutated causes rare forms of ALS, plays a functional role in how mutant SOD1, which is another ALS causal gene, is degraded in human motor neurons when that gene is mutated. So here's an example of a mechanistic interaction between two distinct genetic subtypes. And last but not least, in work that we published over the last few years, we've shown that two very distinct genetic causes of ALS, C9orf72 and NEC1, which is a kinase that's involved in regulating several cellular processes, impact the same cellular pathway in cells, a pathway known as nuclear import, through very different angles. So this is yet another example of two very different causes of ALS directly affecting the same cellular pathway through different processes. So these are three examples of convergence, mechanistic convergence between this distinct genetic subtypes that we've uncovered over the last few years. So what about sporadic disease, which is what the majority of patients are diagnosed with? And in the lab, we always think about models and approaches we can develop to get an insight into sporadic disease. And for today, I'd like to focus on a new story and I'll tell you about a target that I'm excited about, which concerns sporadic disease. So a couple of brief notes about sporadic disease. So although the disease is highly heterogeneous, both genetically and in terms of clinical presentation, the overwhelming majority of ALS patients, about 97%, share these common neuropathological, this unifying neuropathological feature, which is the nuclear loss and cytoplasmic accumulation of a protein known as TDP43. So TDP43 is an RNA binding protein that typically lives in the nucleus of neurons, and it has several roles there, including splicing and metabolizing RNA molecules. And for reasons for which we do not understand, TDP43 in ALS patients leaves the nucleus and forms these cytosolic aggregates. A lot of people are trying to understand why this happens and what we can do to reverse this process. So we think that this is a very important, obviously, pathological feature. And another unifying clinical feature of ALS patients is this pronounced cortical and spinal motor neuron or spinal neuronal hyperexcitability, which has been recorded by either TMS studies or peripheral nerve conduction studies by several groups over the years. The underlying causes of this hyperexcitability remain unknown. There's debate about whether these are cell autonomous and they're driven by defects in the neurons themselves, or they're driven by lack of inhibitory interneuron networks. But nevertheless, several researchers have shown that these characterize the overwhelming majority of ALS patients, and in some cases, they've shown that these excitability can happen pre-symptomatically. So C9orf72 patients, for example, that have not yet been diagnosed with ALS exhibit this cortical and peripheral hyperexcitability. So we wondered whether these two unifying pathological features of TDP43 mislocalization and this excitability could be mechanistically connected in some sort of way. So this is a simple question we try to address, which is there an interaction between TDP43 and neuronal hyperexcitability? And to begin to address this question, Joseph Klim and Brian Joseph, who were Kevin Egan's lab at the time, did a very simple experiment. They took a human embryonic stem cell line, which has a reporter for human motor neurons. They differentiated these into human motor neurons, fact purified them, so we can only look at these motor neurons, which is a cell type that becomes defective in ALS. And then they depleted these cells from TDP43 by using siRNAs to simulate part of what happens in patients because TDP43 function becomes impaired in ALS individuals. And at the end of this, they did an RNA sequencing experiment, and on top of several genes that become misexpressed, they identified several genes that become misspliced. And we become interested in missplicing alterations as a cause of TDP-43 over the last few years, driven by some of the breakthrough studies identifying genes like STAFMIN and UNC13A as missplicing targets in ALS. And the reason why we're interested in splicing is because sometimes splicing alterations do not necessarily cause changes in gene expression. So there are several genes that we identified as being misspliced, but one gene that caught our attention that I want to talk to you about today is this gene called KCNQ2, which encodes a voltage-gated potassium channel. So this is a specific missplicing alteration that happens in these motor neurons. So at the top, these are sequencing graphs, sequencing tracks. On the top, I'm showing you this is the structure of the gene of KCNQ2. And what you're seeing here is that most of the sequencing tracks go from exon 4 to exon 5 and exon 6. But when we deplete neurons from TDP-43, you see that a proportion of transcripts, about 1/3 of transcripts here, basically entirely skip this exon called exon 5. So you go directly from exon 4 to exon 6. So in stark contrast to STAFMIN and UNC13A, this is not a cryptic event, this is an exon skipping event. This is the molecular validation of what I showed you. These are the SI scrambled neurons and the SI TDP-43 neurons. And what you're seeing here is that when we deplete neurons of TDP-43, you see that a proportion of transcripts basically lacks exon 5, and you see that down there. And this is the quantification. This is a significant enrichment of this exon skipping event, about 40, 50-fold in neurons that are depleted from TDP-43. We know that this is a TDP-43 dependent event because we see it when we deplete neurons from TDP-43. But to prove that this is actually directly caused by TDP-43, what we had to do is perform an RNA immunoprecipitation experiment and show that TDP-43 actually binds the KCNQ2 transcript directly downstream of exon 5. This is the quantification of the immunoprecipitation down here. You can see the enrichment for TDP-43 binding directly downstream of this exon, exon 5. So we think that this is a TDP-43 dependent effect. So why I'm excited, why am I telling you about this KCNQ2 channel? This is a voltage-gated potassium channel that regulates a very particular type of current in neurons known as M current. This is the structure of the channel here. And in very simplistic terms, activation of this current prohibits neurons from firing repeated action potentials, while inhibition of these channels allows neurons to fire trains of action potentials, so continuously firing. And in my lab, we've been interested in this channel for a number of years because dominant negative somatic mutations in KCNQ2 are known to cause very early onset seizures. And we've been studying these processes in a whole separate project in the context of epilepsy. So obviously ALS patients do not have seizures, but as I will explain, we're talking about basically a somatic mutation versus a proportion of a patient's brain now exhibiting potential dysfunction in this KCNQ2 channel. So does this happen at all in ALS patients? Because what I showed you so far was human motor neurons grown in vitro. So to begin to address this question, we utilized the Target ALS New York Genome data sets. This is the largest ever assembled sample set of post-mortem ALS tissue. This is bulk RNA sequencing that's been done from various different tissues from the central nervous system, from ALS patients, controls, and neurological controls. And it's freely available. Anybody can download it and use it. And to define whether this event happens in ALS patients, we followed a simple strategy. We asked whether we will see any evidence for this novel exon 4, exon 6 junction in postmortem ALS tissue. And what I'm showing you here at the bottom is that when we looked at tissue from 918 ALS patients versus controls, we see a very highly significant enrichment for this exon skipping event, specifically in ALS patients. Not only do we see this, but this correlates well with cases where we would expect to have concomitant TDP-43 pathology, like C9orf72 cases or sporadic cases, but we never see it in cases where we do not have TDP-43 pathology, like SOD1 patients or FUS patients. So this is how the abundance of this exon skipping event looks like in the CNS. So we see this in about 30% of motor cortex cases that we've looked at, and we also see it in about 17% of lumbar spinal cord cases, whereas tissues which are thought to be less involved in ALS, like the occipital cortex or the cerebellum, we see significantly less proportion of cases where this is detectable. And this is the significant enrichment specifically within the motor cortex of ALS patients. In this particular case, we've got 269 patients versus 37 controls. And hopefully what you can see here is that this is highly enriched for ALS cases. Now, to get some relevant information about the concordance of this with TDP-43 function, we utilized another data set, which was generated by A.D. Lee, where he was able to fax purify from postmortem ALS tissue, specifically from the frontal cortex, neuron-positive neurons that had very high levels of TDP-43 or neuron-positive neurons that had very low levels of TDP-43. And the beauty of this data set is that we can look at individual patients. Here I'm showing you six cases within their TDP-43 high and TDP-43 low expressing cells or nuclei. And on the left, I'm showing you that we never ever see exon skipping for KCNQ2 in the TDP-43 high expressing neurons. But in those same patients, we always see exon skipping in the TDP-43 low expressing neurons. And when we quantify the percent of transcripts that exhibit this event, we see that it's about 50%. So here on the left, I'm showing you exon 5 inclusion in the TDP-43 high neurons is basically 100%, but exon 5 inclusion is only seen in about 50% of transcripts in the TDP-43 low cells. So again, I showed you that we discovered this in human stem cell models. We validated this happens in patients, and we validated now that this correlates strongly with TDP-43 in postmortem ALS tissue. So does this event correlate at all with disease metrics? Is it relevant? To address this question, we teamed up with Len Petruccelli and Mercedes Prudencio at UMass, who had another beautiful data set of ALS FTD cases, about 89, I think, and 27 controls. We designed specific qPCR primers that would specifically look at this exon skipping event. We sent these primers to Len, and when they run their analysis, they're able to show that this basically is an ALS FTD specific event. We never see it in any controls, and we see highly significant enrichment for this event in these ALS FTD cases. Now, the beauty about this sample set is that Len had concomitant corresponding TDP-43 pathology as well as clinical information about these individuals. And what I'm showing you here is a simple correlation graph showing that the abundance of this KCNQ2 exon skipping event highly strongly correlates with the amount of phosphorylated TDP-43 in the frontal cortex of these individuals, again suggesting that this is a TDP-43 triggered event. What's more excitingly is that when Len looked at the correlation between the abundance of this effect and the age at onset for the disease for this individual, there was a very strong inverse correlation shown here, which is all that means is that the younger somebody was when they were diagnosed with ALS, the higher the abundance of this exon skipping event was for KCNQ2, and conversely, the older somebody was when they were diagnosed with ALS, the lower the abundance of these exon skipping events, suggesting that this correlates well with disease metrics, and perhaps suggesting that it might play a role in disease onset. So how does this specific event affect the function of the channel itself? And ultimately, the function of the neurons was the next question we want to tackle. So this exon 5 encodes the entire channel pore of the channel, the pore of the channel, and parts of the transmembrane domain and part of the pore loop. And I should point out that unlike other missplicing events, this does not lead to a premature stop codon. So we've got basically an irregular protein that's translated at the end of this event. So our in silico analysis predicted that we would basically come up with sort of weird protein that's translated, perhaps misfolded, we're not sure at this point. So to address how the function of the protein would be impacted, we teamed up with Al George and Carlos Vanoe, and we did a traditional heterologous expression model where we take a channel, KCNQ2 channel, without the mutation or with this particular exon skipping event, and we asked how much potassium carnate is it able to conduct. And to cut a long story short, what I'm showing you here is a positive control on the left, which is a wild-type channel. And the mutant, the exon skipping event basically results in a dead channel. It's a channel that's not able to conduct any potassium current. And the effect of this is similar to a lot of mutations that cause very aggressive early onset epilepsies in children. So this was a heterologous expression system. How do we model this specific event in human motor neurons, but in the absence of TDP-43? To address that, we employed CRISPR-Cas9 gene editing strategies and introduced homozygosly this exon skipping event in a human embryonic stem cell line. And then we took that human embryonic stem cell line and its isogenic controls and made cortical neurons this time. And what Kelly Marshall was able to show when she examined these neurons is that in contrast to the control neurons, when we look at the endogenous KCNQ2 channel in these cells, we see that it basically accumulates within the cell body. It accumulates what we think is within the endoplasmic reticulum. This is shown here in this red channel here, and it doesn't get to the membrane. So we think this is why this channel is probably dead and unable to conduct any current. This is some beautiful staining that Kelly was able to do to show that in the cases of the mutation, the channel is entirely enveloped within calnexin here, which is a marker of the ER. So in contrast to what happens to the wild type channel, which makes it over to the membrane to control excitability, this exon skipping event results in this channel getting stuck within the ER. Does this happen in patients? So again, we started from motor neurons, we went to patients, went back to cells, now we're back to patient tissue. And what I'm showing you here is postmortem spinal cord tissue. This was staining done by Jacob Mann in the lab. This is a large spinal motor neuron in healthy controls. TDP-43 is in the nucleus. And basically, this is what KCNQ2 staining looks like in these cells. But in contrast, in sporadic ALS patients which lack TDP-43 from the nucleus, we see this focal accumulation of KCNQ2 within the cell body, similar, but not exactly the same to what we saw in this in vitro models. Now, Jake was able to show that these accumulations are actually ubiquitinated, shown here by the co-staining of KCNQ2. And again, this is endogenous KCNQ2 in postmortem ALS patients, suggesting that these are probably aggregates of KCNQ2 that are ubiquitinated. So, so I showed you that again, that this happens in cells, it happens in people, it happens in patients, correlates with disease metrics. But what I haven't shown you yet is how are neurons affected, and specifically their excitability. So to do this, we're in the course of doing several experiments. I'm just going to show you one brief set of data where Kelly Marshall plated these stem cell derived neurons on these multi-electrode arrays that record spontaneous activity from any neural subtype that you played on them. And the beauty of this is that you can collect a wealth of information over several weeks in culture. And what I'm showing you here is that what Kelly found was that several firing metrics, including bursting and several metrics associated with this bursting activity, are significantly increased in neurons that basically have this exon, this KCNQ2 exon skipping event homozygosly engineered within them. Again, suggesting that these neurons become hyperexcitable when you have this specific, what we think is a TDP-43 dependent dysfunction. So what we think is happening, but we need to continue to build up the proof here, is that for the first time, we're able to connect TDP-43 dysfunction or the unifying pathology in ALS patients with a clinical presentation of cortical and perhaps peripheral hyperexcitability through this KCNQ2 exon skipping event, which is a TDP-43 dependent event. We think that this is driven by both gain of function events, because this channel gets stuck in the ER, as well as loss of function effects, because this channel is dysfunctional, and we think that this leads to hyperexcitability. By no means do we think that this is the only event that drives this hyperexcitability, but we think it's a major contributor. So if that's the case, we next started thinking about how can we rescue this defect to basically test causality. Is this important for what happens in these cells? And to do this, we decided to design splice modulating antisense oligonucleotides. We'll hear a lot about this during the keynote presentation today. But what these molecules do in this particular case is basically simulate TDP-43 binding within mRNAs. And to do this, Hao Chi, a postdoctoral fellow in the lab, collaborated with Jonathan Watts out of UMass, an RNA biologist, and they designed a set of antisense oligos that basically sit immediately 5' and 3' and 5' of this exon 5. And Wanhao was able to conduct a screen, a small level screen, where she took wild-type human motor neurons, depleted them from TDP43 to simulate TDP43 function, and then assessed the ability of these antisense oligos to restore this KCNQ2 missplicing. And to cut a long story short, Wanhao was able to identify several molecules that are able to restore this missplicing event. This is one of our best molecules. It's able to restore it by almost 90%. And we're in the process of assessing the efficacy or the ability of these molecules to rescue excitability and other effects. But for the last minute, I want to say it's not obviously just KCNQ2. We know that there are several other transcripts that are misspliced as a result of TDP43 dysfunction. And Matt Wright and Francesco Alessandrini in my lab have been utilizing target ALS data sets to filter and prioritize what we think are the most critically important misspliced TDP43 transcript based on the abundance that we see these in postmortem tissue and based on the biology. And as an example, what I'm showing you here is that the motor cortex and the spinal cord are the most affected tissues in terms of differential expression in ALS patients. And TDP43 or TDP43 dependent missplicing is most abundant, again, in the spinal cord and the motor cortex of these individuals. And as a start, we've nominated 3 TDP43 missplicing events, STATHMIN, UNC13A, and KCNQ2. And in collaboration with Chad Merkin's lab, we're trying to come up with ways that will allow us to co-deliver multiple splice modulating ASOs into neurons at the same time. This is one example where we're using spherical nucleic acid technology to either put these ASOs on the surface of these lipid nanoparticles, or we're starting by putting them basically inside the lipid nanoparticles and co-deliver these events simultaneously on neurons and then assess the ability or the efficacy of these molecules to rescue neuronal excitability. So I'll summarize there. I've showed you just a little snippet of how we're continuing making progress on characterizing mechanisms in rare genetic subtypes of ALS. And what we're learning so far is that every time we've looked at a genetic subtype of ALS, we're seeing some interesting connection with another subtype of ALS. I've told you about how I'm excited about this KCNQ2 target, which we think is highly relevant for sporadic disease. And last but not least, I've showed you how we're making efforts to simultaneously target several TDP43 missplicing events like KCNQ2 to assess potential therapeutic efficacy. So I try to mention the people that led the work throughout the talk. Kelly Marshall, with contributions from Dina Simpkin, Jacob Van, and others, have been leading the KCNQ2 project. Brian Joseph and Kevin Egan are collaborators on this. And here I have several other collaborators with who we have exciting projects ongoing in the lab. I have several posters out there. Hopefully you'll get a chance to take a look at them. And I'd like to mention the New York Stem Cell Foundation and Les Turner Foundation have been supporting my work this 8 years that I've been here. So thank you, and I'd like to take questions if there are any. Thank you.
Yes, for questions in the audience, please use the microphones we have on the aisle. And for people who are watching through the Zoom link, I am keeping an eye on the chat. And if there are any questions, I'm going to read the questions on the chat. But first, please, the questions in the room.
Hello, that was really nice. I particularly enjoyed your framing of the convergent pathways and how studying one gene reveals a connection mechanistically with others. So along those lines, in your cells in which you express the version of KCNQ2 that can't be spliced properly, so you cannot have these, so you get these trains of action potentials.
Right.
In that cellular model, have you guys looked to see whether then the cells also develop this proteostasis problem of like accumulation of the KCNQ2 protein?
Yeah, that's a great question. So in these homozygously edited cells, we do see this accumulation of KCNQ2. And the only other thing that we've done to partially address your question, when we look at the gene expression profile of these neurons, which only have KCNQ2 dysfunction, and ask whether it looks similar at all with what's seen in postmortem ALS tissue, we're seeing a similar or a conversion, a correlation in the gene expression profile of these cells with the gene expression profile of what happens in postmortem ALS tissue. But we're not entirely sure what's that driven by. Did I get that?
So do you mean signatures like that would indicate that the cell is struggling to handle misfolded proteins and so on?
Well, we're not seeing a lot, surprisingly, we're not seeing a lot of that. We're not seeing any clear signs of what we would expect to be ER stress, but we're seeing, for example, several heat shock proteins or one or two heat shock proteins.
So probably they're handling it because it's just the one hit. It's really interesting.
Right. And it could be that if it's one hit, they're able to handle it and survive, but we're not sure exactly how that correlates in the context of TDP-43 dysfunction.
Oh, thank you.
Evangelos, really beautiful work. My question I have is, if you induce hyperexcitability, do you see missplicing of KCN2?
Yeah, that's a great question. I don't know that we've done that experiment well, but the answer would be no, we don't see that.
Okay.
But we're really interested in getting to the bottom of this. We're hypothesizing that there might be some sort of feedback loop between one missplicing event and others. So, but yeah.
And just a follow-up question on the demographics of the patients. Do you know if there's any correlation whether they experience this cortical hyperexcitability in the presence of this missplicing event? And can you get at that with some of lens data?
That would be a very informative question to ask. We just don't have the sample set that would allow us to do that. The only exception to this would be the patients that participated in the retigabin trials. But I don't know that we have access to postmortem material from these individuals. But that would be highly informative to ask. Yes, absolutely. Yeah.
So I'm not a scientific person, so I'm going to ask the question, and I'm not sure if it's a question that I should be directing to you or somebody else, but I have genetic ALS. And so my question is, that being the case, does ALS that early on cause developmental problems during pregnancy? For example, I was born with Hirschsprung's disease. So would that be, and which, for those of you who don't know, that means part of your intestines are basically dead and didn't form correctly. So is there a correlation between genetic ALS and some kind of developmental issues during pregnancy? Or has that even been thought about at this point?
Yeah. So as far as I'm aware, and I would invite other researchers and clinicians to correct me if I'm wrong, there hasn't been any definitive association between genetic ALS and neurodevelopmental problems to this point, at least. Yes. We think that this is an adult onset disease that doesn't affect development. Yes.
Very nice talk. Thank you so much. Two quick queries. First, are these potassium channels relegated to the cell soma, or do we see them out in the dendrites as well? And secondly, do we need to develop better diagnostic tools to identify hyperexcitability in ALS so that we can implement these therapies at the right time? Because I feel like that at the point where it gets diagnosed is too late, sadly.
Yeah, both great questions. So to address your first question, so this particular KCN2 channel is typically found within the AIS in neurons. That's where it's predominantly localized. And I didn't have time to show you this, but if we grow our stem cell-derived neurons long enough in culture, we see focal accumulation in the AIS where we would expect to see it in the at least healthy control neurons. But again, in this homozygously engineered models that we've created, KCN2 does not make it there. It gets stuck in the AR. As far as the second question is concerned, absolutely, we need better biomarkers of excitability. I think this TMS studies, have been incredibly informative. But again, I'm not a clinician. I'm not privy to the exact execution of this. But my understanding is that they're complicated to execute in the context of different places. So there's been some discordance between data generated from one side to another. So I think there's always interest in developing better biomarkers of excitability. So, but yeah, that's outside our areas of expertise. Yeah.
Oh, hi. So beautiful data. My question is, KCNQ2 is quite ubiquitous, right? Expression. So do you have any data or any hint about why would it affect selectively some neurons and not other? I'm thinking of interneurons, for instance. What do you know about possible problems with the expression of KCNQ2 in interneurons, and might this affect excitability through inhibitory circuitries?
Yeah, that's a great question. So we think that this is driven by TDP43 dysfunction. So TDP43 accumulation is thought to be excitatory or motor neuron specific. So as far as I'm aware, there's not a lot of data to support the idea that there's TDP43 dysfunction in inhibitor interneurons. So if that's not the case, then we wouldn't necessarily expect to see KCNQ2 dysfunction. So we think it correlates with that. But that's to be determined, because I think there's this growing sense in the field that perhaps you might have some level of TDP43 dysfunction in the absence of clear-cut cytoplasmic accumulation. So as we're collecting more data and from postmortem tissue using various different approaches, we'll get to the bottom of that. But lack of cortical inhibition is one of the theories that's been put forward as to what might be driving this excitability. Yeah.